Hello, my name is Innocent Sangiumva, and I'm a junior here at Concordia. I'm a Spanish major with minors in psychology and chemistry. Back in 2019, I took behavioral neuroscience with Dr. Olson, and I really enjoyed the course, and so I asked him about joining his research team. And so I applied, and I worked over the summer with him in 2020. Dr. Olson is a professor of psychology and neuroscience, and his research focuses on learning and memory formation. So back in early spring of 2020, Dr. Olson applied for funds from summer research endowments and received some money for us to complete a research project. So the original plan was for us to continue the research from the previous year and to do more experiments in the lab. But purchasing the rats and shipping them and quarantining them and then getting them acclimated to us is a process, and it takes a lot of time. And so with the uncertainty of a return date to in person and just the threat of being shut down at any time led to a change in the project. So we decided to alter the plan to a grant writing proposal. And so that entailed us reading current literature, collecting data from the previous year, and planning future experiments. And so this poster here is a good visual representation of the focus of the grant proposal, and it also highlights the direction in which we hope to move forward. Start here with some of the general background here. So memory consolidation is a phenomenon that enables processed memories to be strengthened and easily retrieved. When we retrieve our memories, they are subject to similar molecular events that occur in the initial consolidation of memories, and thus can be strengthened and fortified. However, in diseases such as Alzheimer's, there is noted failure in this memory retrieval step. In order to investigate the memory retrieval and reconsolidation processes in the brain, animal subjects such as rats can be trained in spatial memory tests such as the Morse water maze. And the animals are tasked with learning to navigate their environment using spatial cues and tasked with locating a platform submerged in the water. Following their learning period, behavioral, pharmacological, and neurobiological aspects of memory can be investigated. Yeah, so this here is the Morse water maze, and it's a tank that's divided into 4 quadrants. And in one quadrant, just below the surface of the water, lies the platform. And so the rat would be released from different locations and would be tasked with navigating the maze and finding the platform. Variables such as speed and time spent in each quadrant can be used to assess learning and during the training process. So one of our specific aims of the project moving forward is we want to further assess the role of the forebrain cholinergic system in memory retrieval and ongoing memory integrity. The cholinergic system has been identified as a critical neurochemical pathway for learning, alertness, and encoding of sensory information. The cholinergic system is active in regulating synaptic membranes and facilitates neuroplasticity in the hippocampus. In normal aging, impairments in learning and memory are observed in individuals as a result of lost cholinergic functionality. And so many prescribed treatments and therapies for impairments are linked to cholinergic agonists. They promote this pathway. In contrast, inhibition of the muscarinic acetylcholine receptor mimics the natural loss of these cells that occurs naturally during healthy aging and leads to the impairments of learning and memory that are observed. While numerous research efforts have aimed to identify the role of the cholinergic system in the encoding of memories, few studies have aimed their focus on the effects of this system on memory retrieval, and that's what we plan to do. So in previous research, Dr. Olson's lab has shown that animals that have learned the water maze task, cholinergic blockade in the hippocampal region impairs spatial memory retrieval. So by day 6 of training in the water maze, rats had found the platform in just under 10 seconds as compared to 45 seconds on day 1. There was significant improvement over repeated trials as the rats had learned to navigate their way around the maze. Rats that received the acetylcholine inhibitor, a drug called scopolamine, did not learn. They showed impaired learning. And this figure here shows the probe time And the probe time is the time spent in each quadrant when the platform is removed. And as you can see here, the probe time is lower for the rats that received the scopolamine. And this here is the traces of the rats and where they spent their time. And so this one here, as you can see, it's centered around here in this kind of in this quadrant here. And whereas in the rat that received the scopolamine, the acetylcholine inhibitor, right, their movements are kind of scattered, as you know, they're not able to find where that platform was. Also would like to further determine the role of extracellular matrix proteases known as matrix metalloproteinase 3 and 9 in memory restabilization following memory retrieval. Matrix metalloproteinases, or simply MMPs, have been suggested to play a role in synaptic plasticity. Upon extracellular matrix degradation, synaptic plasticity allows neurons to reorganize and modify the structure of existing connections, a process that is central to the formation and consolidation of memories. Interestingly, inhibiting the function of these MMPs during learning impairs the ability to learn, suggesting that these MMPs, specifically MMP3 and MMP9, are required for the consolidation process. While MMPs are recognized as key players in forming memories, their specific role in the retrieval and reconsolidation of memories is largely unknown. Previous results from our laboratory give a preliminary indication that MMP9 levels are increased following retrieval of a spatial memory. In addition, it also appears that cholinergic blockade further increases these MMP9 levels in the hippocampus. So this figure here shows the different MMP9 levels in the different groups. And so the cage control, the learning group, and then the group that received the cholinergic inhibitor scopolamine. And as you can see here, in the group that had the cholinergic blockade, that had the highest levels of MMP9s. And so, and then this figure here is the Western blot comparisons within the different groups. And if you compare the group that received the scopolamine versus no scopolamine, the darker indicates more protein levels. And so the cholinergic blockade of the hippocampus increases MMP9 levels. In our proposed research experiment, 68 animals will undergo 6 days of water maze training, followed by intrahippocampal cannulation surgery. And this here shows where the cannula will be placed here in this region of the hippocampus. And after 5 days of post-surgical recovery, animals will receive bilateral injections of either artificial cerebral spinal fluid or the muscarinic acetylcholine antagonist, scopolamine. 20 animals will receive no further exposure to the water maze and will serve as yield controls for the protein analysis described in specific aims 2. Remaining animals will next be subject to a probe test 1 5 days after the cannulation surgery. After a rest period of 5 days, another bilateral injection of ACSF or scopolamine will be administered and animals will receive another probe test 2. Importantly, a portion of rats that received ACSF prior to probe test 1 will receive scopolamine prior to probe test 2, and the other portion will receive ACSF. In a similar fashion, a portion of rats that received scopolamine prior to probe test 1 will receive ACSF prior to probe test 2, and the other portion will again receive scopolamine. So at this point, all animals will be euthanized and their tissue will be harvested for protein analyses. And this is a good visual here of the timeline. And so starting with training, surgery, and then either ACSF or scopolamine, followed with the probe test, a 5-day delay, followed by ACF or scopolamine, and then another probe test. Research questions that we hope the experiment provides. Does scopolamine at probe test 1 affect the ability to retrieve spatial memories in the absence of scopolamine 5 days later at probe test 2? And does scopolamine prior to probe test 2 impair retrieval of a memory that was successfully retrieved in the absence of scopolamine 5 days prior? And so the animals that from the initial learning phase of experiment 1 that did not undergo the further maze training, together with the animals that received only the first probe test, will serve as the controls in Western blot comparisons against animals from all the other experimental conditions. Their tissue samples will be used to determine answers to the following research questions. Do MMP3 and 9 levels increase when scopolamine is given prior to probe test 2, 5 days after the successful retrieval during probe test 1? Is the magnitude of increase diminished in comparison to animals given scopolamine prior to probe test 1? And does scopolamine at probe test 1 affect MMP3 and 9 levels 5 days after probe test 2 in a way that is different from the artificial cerebrospinal fluid group? Appreciate your time and attention, and I hope to see you at the Q&A.